So, hello, good morning, good afternoon, wherever people are. Welcome to today's Youth Talks, Young Scientists Shaping the Future. Just to very briefly introduce myself, my name is Nako Nakatsuka. I'm a senior scientist at the ETH Zurich in the Laboratory of Biosensors and Bioelectronics, and I also work in the area of nanopores, so I'm super excited to introduce the group of really fantastic young researchers who will give talks today. We will have Mortaza Aramesh, Dennis Garoli, and Radha Boya talking on the topic of biomimetic nanoscale materials. And so I'm super excited that each of these individuals will talk about their research. And then at the very end, we'll have a chance to come back all together and discover some future areas of the research that we'll discuss. And so I would like to start by introducing Professor Radha Boya. Professor Radha Boya, FRSC, is a professor, Royal Society University Research Fellow, and Kathleen Ollerenshaw Fellow at the University of Manchester. After completing her PhD in India, and after a brief postdoctoral stint in the United States, she has secured a series of highly prestigious international research fellowships that have enabled her to rapidly build her research profile in the United Kingdom. Radha has published 60 research papers, including several of these in Nature and Science journals. Radha was awarded an ERC Starting Grant, Analytical Chemistry Young Innovator Award, Philip Leverhulme Prize in Physics, RSC Marlowe Award, UNESCO L'Oreal International Rising Talent, the list goes on, L'Oreal UK and Ireland Women in Science Fellow, and she was also recognized as an inventor of MIT Technology Review's Global Innovators Under 35 list. And so we're very happy to have her today, and the title of her talk is Angstrom-scale Channels Made from 2D Materials, Molecular Transport. And so now the floor is yours, Radha, and very excited to hear your talk.
Thank you very much for your kind introduction, Nako, and I'm very happy to share our research today. I'm glad to speak to you all today. My slides are up. Hopefully they're all okay, seen. Yeah. So briefly, I'll talk about our latest research on Angstrom-scale channels today. So I'm talking to you from United Kingdom, Manchester. So this is our university main building. And we have a Graphene Institute, which is a Center for 2D Materials Research. It's a central institute which is funded by government for enhancing the research on 2D materials. So as you may know, so Manchester is well known for discovery of 2D materials. So the 2D materials discovery is very popular. I mean, starting with graphene, owing to the simplicity of the method of preparation. So you take a layered crystal like graphite and use a simple scotch tape to remove one layer of graphene. Yeah, so which is one carbon atom thin sheet, one atom thin sheet. This discovery was awarded Nobel Prize in 2010 in physics to Professors Andrew Geim and Gaussian Novoselov. So this is all from then on. I think there has been revolution in terms of 2D materials research with several papers coming. It's only increasing every year, right? So what I'm interested is not on graphene, but the opposite, which is graphite. So what if we could remove this layer of graphene, not from the top of the crystal, but from the middle of the crystal? So what we would be left with is a cavity which is of the thickness of the layer removed. So with this idea, we started this research several years ago. So the idea of two-dimensional voids. So what would this 2D empty space offer? So this was the key point where we started this whole research area. So and why are these two-dimensional empty space two-dimensional voids interesting? So mainly due to their dimensions. I would introduce like the fluidics, how the fluidics how the fluidics devices have evolved. So microfluidics as a field evolved in 1960s, so where people started making concurrently with microelectronic devices. So where there were methods to fabricate microelectronic chips. Similar methods were employed for making microfluidic devices. And over 90s and 2000, then the field of nanofluidics started to emerge, wherein the dimensions were scaled down much more. Less than 100 nanometers dimensions were possible to make, and there were even like all dimensionalities, like zero dimensional pores or nanotubes, like which are one dimensional, or slits were possible to make, like two dimensional slits were possible to make. And even soon after the discovery of graphene was also used, immediately adapted for making nanopores, right in 2010. So right now, I would say this is an interesting time wherein we have methods to make even smaller channels. So we can make channels which are 1 atom wide, like 1 atom wide pores, or sub-nanometer nanotubes, or 1 atom thin channels, like how we make. So this is an interesting time where we have methods to make this kind of small systems, and also methods to characterize, such systems. So this, I would say, is Angstrom-scale fluidics era. Hopefully, this continues to bring many interesting, exciting properties that we still don't, that we never saw in nanofluidics channels. So, and at this length scale, so we have completely, ability to confine one molecule to one molecular layer. Let's say if we have a channel which is only allowing water molecules. So such kind of channels exist in nature. For instance, protein channels, which can selectively filter out only water versus other ions, or which are selective to one ion over the other. Such kind of confined channels exist in nature. On the other hand, it is quite interesting to have such channels for sieving purposes. For instance, if you are able to separate out salt from water, so this kind of pores would be quite useful. So with that interest, so we started we started making these kind of 2D channels, which can be of the dimensions of the molecules themselves. So I introduced the concept of two-dimensional voids, but how do we make them in practice? So we make them in practice by a method, by assembly, which is like Lego-like assembly. So we take individual parts and then assemble them by putting them one on top of each other. So basically, this each two-dimensional channel has 3 parts: the bottom layer, which is a 2D crystal, and the spacer, which is a 2D material as well, and then the top. The middle layer, which is the spacer, decides the height of the channel. This could be 1 atom thin to several atoms thin. So this is an example of cross section of one such channel, which is about 2 atoms thin. So bottom and top layers are graphite, and the spacer is also graphene. So we can obviously vary all the dimensions of the channel, the width, the height, and the materials. So we could have a 1 atom thin channel to several nanometers thick channels. So this is an example of 15 nanometer tall capillary. And we can have mix and match surfaces. So the top and bottom walls could be of one material, the spacer could be a different one, or we could have asymmetric channels. So there's a level of programmability, in these channels. So, and all of these 3 layers are held by van der Waals forces. There is no glue which is holding them. They are just held together because of their atomically smooth nature. So there can be van der Waals forces between these layers and which holds them together. So we could have channels walls made from like hydrophobic materials or hydrophilic materials like mica, for instance. So there's a level of high level of we can define what the channel has to be made from. And all these beautiful cross-sectional images reveal, how the interfaces, or these cross-sectional images from Professor Sarah Haig reveal how smooth the interfaces are. So there's between the spacer and between the spacer and the top and bottom layers, the interface is quite seamless. Yeah, so which makes it a leak-tight channel. So we make these sandwiches of 2D capillaries, and how do we measure flows through them? So we need to have a device architecture which enables the measurement of flows through these capillaries. So for that, we assemble these capillaries on a membrane-like structure, like silicon nitride membrane, and this is the bottom layer. So, and this is the spacer layer. We can define how many channels can be there in a given device. So there could be one channel only, or there could be several hundreds of channels. At this step, we can program it, and we create the opening by dry etching methods. And now we place the top layer. So now if you see the sandwich, it's on the top of this substrate, but the entry and exit of these capillaries is separated on either sides of this wafer, the chip. So this is how the chip looks like. So, and this device architecture was key for our further measurements, because it enables just keeping this membrane between two chambers, and we could measure the flows while the liquid enters from one side and exits from the other side. So this is how it looks like in optical microscope. So this is the top graphite, which is quite thick, and it's deliberately made thick so that the top layer doesn't sag into the channel, because the channel, if you see the aspect ratio, it's only 1 to 2 atoms thin, but it's quite wide. It's about 100 nanometers wide. So we need to have sufficient bending rigidity for the top layer not to sag into the channel. And of course, we can also make channels which can deliberately be sagged into and partially be closed for specific experiments. So we also do that. So that is also possible. So, I've told you how we make these channels and devices. So, and what kind of properties have we measured so far? So, primary directions, I would say, are in like gas flow measurements, water flow measurements, and ion selectivity measurements, either by voltage-driven ion flows or pressure-driven ion flows. And because of to keep it in time, so I would go specifically into the most recent studies. So only the most recent studies I will talk about. The first and foremost study that we had done, because to our surprise, we initially thought even nothing would flow into these kind of tiny channels. So when we tested water flow in our first paper, so, and surprisingly, the water can enter through these channels without any other further pressure. So it's natural capillary pressure which can drive the water through these capillaries. So, and the measurement that we had done was quite simple. So we put this chip with the angstrom channels on a container which contains water, and as the water evaporates, we monitor the weight of this container. So it's micrograms loss, and that can give us the estimate of water flow. So the water flow or the weight loss of this container was measured for several channels, several devices with varying thickness. As the height of the capillary increases, is there any relation to the water flow? So what we see is, as the channels are 10 nanometers to 3 nanometers, we see there is a sort of linear relation, and it follows slip-corrected Hagen-Poiseuille equation. Whereas below 2 nanometer channels, there was anomalous behavior. And this highest point, the water evaporation rate, can be up to 1 meters per second, which was quite surprising to see, given the smallest cross-section of these channels, that water can flow at such high speeds, and which also leads to slip lengths about 60 to 100 nanometer, as estimated from the simulations and from the Hagen-Poiseuille equation by fitting the flows. So we established that water can flow through these channels, and the water flow is quite dependent on the surface of the channel. This results are for graphene capillaries. If we do the same measurements with boron nitride capillaries, the results are completely different. The water flow is not as fast, and in fact, there is no slip on boron nitride capillaries. So it is quite material dependent, the property of the water flow. So now that we established the water can flow through these capillaries, so then we ventured into looking at ion selectivities. Let's say if we put salt into the water, is there any selectivity between different salts, or if we can filter out salt between salt to water? So for these kind of experiments, our setup for measurements is quite simple. So we take this silicon nitride chip and mount it between two chambers which contain the salt solutions. So these salt solution can be of the same concentration and are different concentration if it is a diffusion experiment. and then we measure the currents through these electrodes. So if there is a current flowing through, it has to be through the channels, because everywhere else it is sealed. So from these electrochemical currents, we can get the conductivity, and we can also get the mobility of the ions from the diffusion if we put two different concentrations. So with these kind of measurements, we measure the ion flows. So just to remember, ions themselves in water, in aqueous solution, they are dressed in hydration shells. Yeah. So when we talk about the size of the ion, it's the ion core size, which is incompressible inorganic core, and there is a water shell around it, which is the hydration layer. So usually when we talk about the size of the ion, it's the hydrated radius that we are talking about in water. So in biological channels, there are channels which can selectively filter out only water from ions, or even those ions which can be selective to one ion over the other, like K plus ion or K plus channel. Potassium channel is very selective to potassium. There are many factors which control this selectivity in biological channels, and their selectivity is also very high, up to few orders. So out of the many factors, at least a few key factors are the geometric confinement and the makeup of the wall, like how the functional groups are arranged. So in our case, we have at least a very good control of the confinement dimension. So we don't have any functional groups. It's a very pristine channel. So we can disentangle what is the effect of confinement in selectivity. So there is no there's almost, these are pristine 2D materials which have minimal defects, which leads to the control of the, which leads us to study the effect of geometric confinement. So in previous studies, we showed that when the channel is very small compared to at least half the size of the ion, we can exclude them. So in the recent study, what we wanted to see is if the ion is able to enter the channel. So that is the size of the ion, which is including the hydrated diameter, is close to the size of the channel. Is there any selectivity between the ions? So for this, we selected several ions which have similar hydrated diameter but differing core diameter. So we have several ions ranging from larger core diameter to very small core. And in this case, is there any selectivity between ion to ion? So with our electrochemical measurements, we calculated the ion mobility. So from the relative mobility, that is Angstrom channel mobility, ion mobility in the channel versus ion mobility in the bulk. If there is no change because of confinement, it will be 100%. Yeah. So there are some ions which have no change in their mobility, or very small change in their mobility when they are in the channel, and there are ions which have completely suppressed mobility. So it so happens that these ions which have suppressed mobility are the ones which are with larger ion core. So, and these, just to remember, there is no resistance for these ions to enter. So that we also confirm by varying the length of the channel. So all of them are able to enter the channels, but once they enter the channels, something is preventing them to have differing mobilities. So to understand this further, we asked our theory collaborators to calculate how these ions behave inside the channels. So this is an example of two ions wherein both are negative ions, fluoride and chloride. So their water arrangement is similar. So in our channels, there is a space to accommodate only 2 layers of water. So because these channels are 6.8 angstroms, so there is only space to accommodate 2 layers of water. So in these 2 layers of water, if we put these ions, what we see is the ion with the smaller core can stay mostly in the center of the channel, whereas the ion with the larger core tends to stay mostly near the walls. So the probability of finding the ion is very high towards the walls when it is larger core. And like this, if we compute the probability for all the ions that we have, the larger core ions like chloride spend most of their time towards the walls. That means the probability wise, they spend almost, when they travel through the channel, they travel along the walls. And only exception here is a lithium. So even though it has very small core, it still tries to spend most of the time near the walls, which we do not understand why it is so, but this is our observation. And there is a correlation between the ion mobility suppression versus the ion spending time near the wall. So from our study, now what we understand is those ions which have very large core tend to travel along the walls, have some interaction with the walls, and that leads to their mobility suppression. So that leads to selectivity between the ions, which are close in size, but still there could be difference in the selectivity based on their inorganic core size. So this is something which is often ignored in the literature. So I think this is something we observed, that it's not just the hydrated radius or hydrated diameter which is important, but also the inorganic core size. So with that, I would now switch gears and go to another recent study that we have published with respect to ionic flows, but with a different application. It's mostly about the ionic memories. So here, what our intention was to see if we can use these ionic flows, ions inside the confinement, to create ionic memories. So just to compare how memory works in human system, it's completely different from how it operates, how it is for a computer memory. In computer memory, it's transfer of electrons, whereas in the human brain, it's transfer of the neurotransmitters, which are again triggered. So it's a chemical reaction triggered memory, and this is triggered with the ion channels. There is an ion channel behind which induces this neurotransmitter motion. And there are several ions, so you could have you could have memories induced with several ions. So there's more variety to the human memory compared to the computer memory. So this is our collaborative study. So basically, in this study, so we collaborated with a group in France, Professor Lideric group. And so what we studied here is looking at the ionic currents, but with much higher voltages. So we now go until like 0.5 volt to 1 volt and look at how the ionic current response is. So firstly, we see there is a nonlinear increase in the current in the capillaries, and when we trace the voltage back, there is a hysteresis, and this happens at both quadrants. If we plot this in terms of conductance, we can see there is at least 2 levels of conductance here, and the hysteresis leading to pinched loops. So this is a signature. The pinched loop with hysteresis is a signature of a memory resistor, memristor. Yeah. So, and the area of this loop is something as a metric we can take to understand the emergence of the memory. So at single point of voltage, you have 2 resistances or 2 conductances, right? So it's 2 states of memory at single point of voltage. So how does this memory come about? So we first important metric we tried was looking at the confinement. So we increase the spacer height. So we make very thick channels, like 90 nanometer thick channels to 0.7 nanometer thin channels. So what we see is, as we go towards very thick channels, there is a linear curve, whereas the thinner channels behave more give us hysteretic curves. So the area of the curve is the area here of the loop can be plotted versus height of the channel, and we see that the thinner channels have higher area and also higher nonlinearity. That means you need thinner channels to get the memory. And not only that, we can also vary the concentration of the salt solution. So as the salt solution concentration increases, it leads to a better hysteretic loops. So we also need more number of ions in the confinement to observe the memory effects. And importantly, these hysteretic curves are function of the voltage frequency. So if we do a sweep of voltage frequency, so we can see that there is a dependence on the voltage frequency. The slower the voltage, the smaller the voltage frequency is, the higher the hysteresis. So all these point to, all these kind of validate that there is a memory within our channels. And why does this happen? So if we look at bulk water versus confined water, which is solubilizing our ions, there is ions in water, right? So in the bulk water, it's a universal solvent, right? Because it has very high dielectric constant. It can solubilize several salts. Whereas in confined space, the dielectric constant is much reduced. I mean, this has been proved also with one of the experimental studies with our collaborators, where they scanned with the scanning probe microscopy, and they observed that within our channels also they observed that dielectric constant can be much smaller when it is in confinement, when water is in confinement. So which leads to increased Zerum length, which means even though ions are very far apart, they can still interact. So the interaction length between the ions, interaction will be much much larger, which can lead to the formation of pairs. So when we are applying the voltage, we are converting these kind of pairs to polyelectrolytes, and these polyelectrolytes are highly conductive compared to the pairs. And the association and dissociation rates of these pairs to polyelectrolytes is not the same. That could explain why this hysteresis occurs in our devices. So of course, and I mean, this existence of polyelectrolytes is not something we can prove, but this is our speculation why this happens, why our memory occurs. So with our current understanding, that's what we think. And practically, can the device remember? So what do we mean by remembering the previous state? And can we write, erase, or modify? These kind of basic, memory characteristics can we show? So this we do by simply applying voltage pulses. So this is the device conductance. So voltage applied here is in blue line and current observed is in the orange line. So if there is no memory, so as long as we repeat the same voltage, we should see the same current. However, we see that for the same voltage, there is a increase in the current. That means there is a history of voltage pulses. This pulse is not the same as the first pulse. So there is a remembrance of the previous history. And this is more easily plotted here. The conductance percentage increasing about from 100% to 300%. So 3 times increase. And erasing also given by the subsequent pulses. And this is the basic demonstration that there is a memory. So we now ventured into advanced learning mechanism. So to put it in simple words, let's say if you hear me only once today, you may forget what I told you in like 2 days, 3 days, right? But if you have seen me several times in several conferences, you may remember. Similarly, if you are learning something, let's say you are learning how to ride a bike or how to play some instrument like piano, you may remember it if you are consistently doing it. So this learning, how it works in human memory is that the time period between the events. Let's say if you are learning something, that's an event, and how frequently you do this event decides how much memory is retained. So this is a plot in a natural neuron. This is from another paper. So basically the conductance change, that's like event, and this x-axis is the timing between the events. So as there is larger timing between the events, the conductance percentage goes to 0. That means there is no retention. But for very small difference in the events, you can see there is a higher retention. So we tried to adapt, this logic and created an algorithm to create an event and looking at the memory, our learning, So this is our event. This is our event to create certain conductance percentage change. So we created a 20% change and read the, if we repeat the events quite sooner, the memory is retained. But if we repeat the event, after a longer time, then the memory is lost. So this learning mechanism is called Hebbian learning. So we can demonstrate Hebbian learning within our channels. And with that, so I would say we have evidence of ionic memory with our channels, and we can do several other things with the confinement that is offered in the Angstrom-scale channels. So which I'm not going to go into detail, but we can do voltage gating, like similar to Similar to ion channels, we can have pressure-induced currents which are voltage-gated, and this could lead to a transistor-like effect, wherein ionic mobility can be modified, or we could put in biomolecules like DNA, or we could have even fluorescence under confinement. This is from the quantum emission of the HBN defects, which are within the channels. So these kind of several studies are possible with our channels. We can adapt our geometry to study interesting things. So I hope I've convinced you that we have a platform which can confine one molecular layer. And I'd like to thank all my collaborators, without whom this work would not have been possible. Several works would not have been possible. Professor Andre Game, Professor Lideric Bouquet from France, and Professor Alexandra from Switzerland, whom we collaborated on the last work that I couldn't talk about, the quantum emission. Professor Sarah Haig from Manchester again. Professor Kees Decker, whom we collaborated with about the DNA translocation. My theory collaborators, Feng Shao and Mehdi, and my colleagues whom I have closely worked with, Ashok, Gopi, Ali, Timothée, Vaine, and Paul, and Theo and Nathan. And this is my group. So thank you so much for all your attention. And yeah, I'll be happy to take any questions if the time permits.
Thank you so much, Radha, for a fantastic talk. I really personally enjoyed it very much. So I see that there's a question in the chat, so I will read the first one. It says, "Thank you for your wonderful talk. I'm wondering, can you control the channel or array on or off?" So maybe in terms of the flow, if you can stop and make it flow again.
Yeah, this is a very interesting question, and it will be fantastic if we are able to have that control. So one way to control it is probably by collapsing the channels intentionally so that they are off to start with, and somehow with some flexibility we can switch it on. Otherwise, these are always in the on mode. So to switch them off, maybe one can do gating with the top layer. So far, we didn't have such studies, but this is a very interesting thing to pursue.
Great. And I don't see any other on the chat, but maybe I have a quick question regarding your experiments of calculating the ion mobilities, and then you compared it to theory. How do you actually calculate the mobility experimentally?
Yeah, hopefully, I have a slide here. Yeah. Okay. So basically what we do is we do the diffusion measurements and we get the membrane potential. So in this similar experiment we do, and the IVs will not be going through the origin, they will go through, there is a membrane potential because of the net diffusion. That membrane potential we can put into Henderson equation to give rise to mobility ratio. And mobility ratio will be between positive ion and the negative ion. And from that we can calculate, in conjunction with the conductivity, we can calculate the individual ion mobility. I wish I had a slide to explain this, but I'm sorry. But this is hopefully I have convinced you how to, I've explained to you how to,
Absolutely. And maybe just a quick follow-up, for the sake of time, we have maybe one more, but you said that it's van der Waals interactions for the stacking of the layers. When you integrate them into these devices and work in high salt, is there ever any issues with the layers peeling off or having some kind of defects form?
Yes. So basically, when we make these devices, not all of them will work. So usually there is, they can sustain high voltages and high salt concentrations up to, like, let's say 1 volt. We have put in 1 volt with aqueous salts because anyway 1.2 the water will start to split. So with respect to concentrations, we have gone up to 2 molar, 3 molar salt concentrations. But in general, the device yield is about 30 to 50%. In a batch, if we make like 10 devices, there could be some devices which are not open because of hydrocarbon contamination, or there could be some devices which give up as you do the measurements. So the top layer especially the top layer is the thickest, so that can come off if you subject it to high voltages. So that is possible. So there is a compromise between, like, how much yield of devices you can get.
Mm-hmm. Great. Thank you so much for your answers. There seems to be no more questions, so maybe we move on to our next speaker. Thank you so much, Radha, and we'll bring you back later for our group discussion. And so
Thank you, Nako. Thank you, everyone.
So now I will share our next speaker of today. I'm very happy to introduce Morteza Aramesh. He received his PhD in biophysics from the University of Melbourne, Australia, where he studied functionalization of nanomaterials for biomedical applications such as single DNA sensors. He was a Marie Curie postdoctoral fellow at ETH Zurich in Switzerland, where he was working on biosensors development for single-cell profiling. He held a group leader and lecturer position at ETH Zurich, where he and his team studied mechanobiological cues to engineer cellular function in immune cells for immunotherapy applications. And this is actually when I met Morteza, and so I'm very happy to have him on here because we are former colleagues. And he is currently holding a tenure-track assistant professorship at the Biomedical Engineering Division of the Uppsala University, working on 3D biomaterials development for engineering cellular response in immune cells. So today, Morteza will give a talk on material and device designs in biomimetic polymer electronics. So now the floor is yours, and we're looking forward to your talk.
Please unmute yourself.
I hope everything is working now. Thank you, Nako, for the presentation or introduction. Good afternoon, everyone. It's a pleasure to be here today and talk about my research. I wanted to give a big thank to Nako for organizing this discussion on biomimetic nanoscale materials. It's a fascinating topic that has a lot of potentials in different fields. Radha's presentation about how molecules move in tight spaces was really interesting and got us thinking how cells behave in similar situations when they are confined in tight spaces. So the title of the talk is Nano-engineered materials to advance CAR T cell production for application in immunotherapy. I'm excited to share what I've been doing and hear your thoughts and the discussion afterwards. Here is the outlook. We will cover 3 topics: engineering cellular response with biomaterials, T cells in immunotherapy in general, and engineering T cell functions with biomaterials. Let's start off with the idea of engineering cellular response with biomaterials. Basically, mechanics and life are linked in many ways. Physical forces have an important role in biology, affecting our tissues and cells in different ways. And if we focus on cells, which are the building blocks of our life, we see that genes and the environment that they are living in both influence the cells, and that determines how the organism appears and functions. But how does mechanics fit into all of this? Well, cells experience various physical forces inside our bodies. These forces have an impact on how cells move, interact, and respond to their surrounding. Think of it like stretching or squeezing a cell, which can influence its growth, migration, and even how tissues and organ function in general. And understanding these mechanics is important because it helps us learn more about biology itself, plus practical applications in areas like regenerative medicine and studying diseases such as cancer is possible. To understand how physical forces impact cell biology, we need to focus on the interaction between cells and biomaterials. This is because biomaterials possess properties that can greatly influence cell function. Various characteristics of biomaterials, including their composition, structure, and surface properties, play a role in shaping the cell behavior. And through customization of the physical and chemical properties of the biomaterials, we can engineer desired cellular actions. For example, designing biomaterials with suitable mechanical properties can mimic the natural environment of the cells, and that may promote the growth and improving the tissue generation. Generally speaking, this field of engineering cellular response with biomaterials holds new possibilities, such as possibilities for new therapies and enhancing the effectiveness of the therapeutic intervention in cancer therapy. Just to give you one example that highlights how the elasticity of a substrate can regulate the differentiation of the stem cells. This is a work from 2006 where Engler and colleagues conducted a study that revealed how the mechanical properties of the substrate on which the stem cells are grown can impact the fate and the type of the cells that they develop into. When mesenchymal stem cells are cultured on a soft and flexible substrate that mimics more like a brain, the elasticity of a brain tissue, something interesting happens. These stem cells have a higher likelihood of differentiating into neuronal cells. The softness of the substrate basically provides mechanical cues that guides the stem cells towards neural differentiation pathway. When the substrate is stiffer, resembling the elasticity of muscle tissue, for example, the stem cells are more inclined to differentiate into muscle cells. Likewise, if the substrate exhibits a higher level of rigidity, for example, like the ones that we find in bone, it provides the mechanical signals that promote the differentiation of the stem cells into bone cells. But this is an example which clearly illustrates the significant influence of substrate elasticity on the fate of stem cells. By providing the appropriate mechanical cues through the substrate, one can effectively guide stem cells to differentiate into specific cell type. Now let's take a look at another important cell type in our body, which are the T cells. T cells are particularly important in the body's fight against pathogens or cancer. Here is a simple demonstration of how the immune system responds to cancer. For example, the cancer here is shown with a gray color in this schematic. The first step involves some specialized cells that are called dendritic cells. They capture the antigens that are produced by cancer cells and travel back to the lymph nodes and present the antigens to the immune system, specifically T cells. And in the next step, protective T cells are generated. And when dendritic cells present this antigen to T cells become activated. They start multiplying and transforming into different subtypes of T cells. And these newly formed T cells specifically recognize and target the cancer cells. They can directly kill tumor cells or activate other immune cells, for example. And the last step is called tumor bed infiltration. And it's the step where the activated T cells interact with the cancer cells, other immune cells, and various components of the tumor tissue. And the infiltration allows the T cell to directly attack and destroy cancer cells. And also it can influence the local immune response. All these steps they can happen naturally in our immune system. For example, it could be that cancer tissues grow in the body, but with this system, body can make it clear and eliminate the tumors eventually. But sometimes the immune response may not be strong enough or may be suppressed by the tumor itself. And that's when therapeutic interventions come into play. Approaches like immune checkpoint inhibitors, adoptive T cell therapy, and cancer vaccines can be used to stimulate or modify the immune system, making it more effective against tumors. And by intervening at different stages, we can improve the process of capturing antigens, enhance T cell generation, and maybe promote better infiltration of the T cells into the tumor, which in general can increase the effectiveness of the cancer therapy. Adoptive immunotherapy is a powerful treatment For some cancer types, it uses patients' own engineered T cells, which are like a living drugs. These T cells are modified to recognize and target cancer cells better. We do this by inserting a gene into the T cells to make a special protein called chimeric antigen receptor, or CARs. And here's how it works. First, T cells are collected from the patient's own blood. Then in the lab, they are genetically modified to express CARs on their surface. This modification helps them identify and bind to specific proteins found on cancer cells, as if it was done by dendritic cells. Once there are enough CAR T cells, they are injected back into the patient's body, and the infused CAR T cells can then attack the cancer cells and, for example, find the targeted proteins and kill the cancer cells. It has shown a great success in certain cancers, for example, blood cancers like leukemia, lymphoma, and especially it's one of the most promising treatments when the other treatments don't really work. There's been a great effort to improve CAR T cell production. Researchers are using different biomaterials with varying properties to enhance the quality and also the quantity of CAR T cells. And by tuning the properties of the biomaterials, one can create an environment that helps CAR T cells grow better and become more potent in killing the cancer cells. So engineering T cell response by tailoring biomaterial means to provide the necessary signals or cues to modulate T cell behavior and optimize the therapeutic potential of the T cells. And this can be done through different ways, for example, by tuning the surface chemistry, topography, stiffness, and porosity of the biomaterials. Recently, we found that changing the surface of biomaterials at the nanoscale level can have a big impact on how T cells behave. When T cells come into contact with the surfaces with nanotopographic features, they become more activated and they multiply at a faster rate. And this can make them more effective in immunotherapy, for example. We also found that nanotopography can make T cells spread out more, rearrange their internal structure, and even have more CAR receptors on their surfaces. And these changes help T cells become more active and work better against tumors. In a conventional setup, for example, T cells are activated with plastic beads, functionalized with activating antibody, and in comparison, the T cells that are grown by materials with nanotopography multiply more quickly and produce more functional T cells. We made an interesting discovery about T cells and how they interact with nanoporous surfaces. Basically, T cells have tiny protrusions on their surface called microvilli, which are important for the function of the T cells. It turns out that when T cells encounter nanoporous surfaces, they start to grow more microvilli, creating the network of tiny extension inside of the pores. And these nanoporous surfaces force T cells to change their shape and grow more microvilli. Generally speaking, the presence of these microvilli is significant because they help T cells to interact better with the surface of the biomaterial. And amazingly, the interaction between T cells and nanoporous substrates goes beyond the formation of these microvilli. We found that T cells exhibit differential gene expression when they come into contact with these surfaces. This means that certain genes, for example, are turned on or off depending on whether the surface is nanoporous or not. And by the way, the image on the left is an electron micrograph of a human T cell on a nanoporous aluminum oxide membrane. And this behavior of the T cells is, we also found that it is pore size dependent among different materials and pore sizes we tested. We found that 200 nanometer pores make a more significant change in T cell activity, for example, compared to other smaller or larger pores. This shows that pore size matters when it comes to how T cells interact with the porous surfaces. specifically pores that are around 200 nanometer size. And the big question here is, why is it so? So when T cells have their protrusions, like microvilli confined within tiny spaces, it has a big impact on the behavior. In confined spaces, especially the ones that are around 200 nanometer in size, an interesting phenomenon occurs that involves the separation of different proteins, in this case, T cell receptors or TCRs and CD45 molecules. As you see in the schematic, TCR is a small protein, but CD45 is a much bigger protein, and they are bound to the cell membrane. What is the role of CD45? It's actually a phosphatase, and it controls phosphorylation of the TCR, which is necessary for T cell activation. And also, it controls other signaling molecules. And the proper segregation between TCR and CD45 can lead to activation of the T cells, because then TCR can remain phosphorylated, and the downstream signaling event leads to T cell activation. When the spaces are larger than 200 nanometer, the TCR and CD45 molecules are not separated anymore inside of the nanopore, and we don't get any activation. Also, when the pores are smaller, for example, around 100 nanometer, it could be very tight for TCR even to get inside of the nanopore. So it's around 200 nanometer pore size that the magic happens. So during this research work, we also found that nanoporous aluminum oxide, which initially seemed to be quite promising, it's maybe not the best choice for making CAR T cells because of its mild toxicity. And to address this concern, we shifted our focus to developing nanoporous polymer structures using biocompatible polymers. Some of these polymers, for example, have been extensively used as implants, biodegradable implants, for example, and show excellent biocompatibility with cells. And we wanted to make sure that they don't harm the body. By using these biocompatible nanoporous polymers, our goal was to create platforms that not only provide the nanopores for T cell stimulation, but also potentially to be used as implantable scaffolds. In these polymer structures, we have control over size, shape, distribution of the pores, and it allows us to optimize how T cells interact with them. With this shift to biocompatible nanoporous polymer structures allows us to comply with the safety concerns during the in vitro production of CAR T cells, which is important for potential applications in immunotherapy. We have made progress in two areas. So one was creating nanoporous polymer scaffolds, and extracting proteins from the extracellular matrix, or ECM, is another area that we are working on. And we want to engineer T cell function using these materials as well. The ECM is basically a network of proteins and molecules that support cells in the tissue. And we have developed methods to remove the cellular components from the tissues and basically achieve proteins that are very close in composition of the native tissue, making it a perfect substrate to studying cell behavior and to engineer T cells. And one advantage of using decellularized ECM is the ability to adjust the mechanical properties of these gels. By controlling the cross-linking and composition of these gels, we can make them more elastic or more like native tissues. This allows us to create an ideal environment for T cells to interact with. These gels are quite interesting because they are printable, they're injectable, and it makes them quite versatile for different applications. They can carry T cells into the body, for example, and release them at specific locations, and they can have a therapeutic impact for a longer term by containing the cells inside of them. So in conclusion, in today's talk, we explored how biomaterials impact T cells engineering and immunotherapy. Here are the key points that I covered. basically biomaterials can influence cell behavior by adjusting the properties like elasticity or surface features. We talked about adaptive immunotherapy using engineered T cells, which has shown promise in treating certain type of cancers. We saw that nano-confinement of T cell protrusions enhances T cell activation and immune response. Also, we talked about biocompatible nanoporous polymer structures that offer safer platforms for T cell engineering. And also, we are now working on printable and injectable gels made from decellularized ECM that can potentially provide an optimal environment for T cells and control delivery. And we hope that more research in this field will lead to more breakthroughs in the future of the immunotherapy. I want to thank everyone who helped make this research possible. In particular, I want to thank my great mentors, Cecilia, Janos, and Viola. My colleagues and students at Uppsala University have contributed greatly to our recent progress, and I'm also grateful for the support of our funding agencies, which has been crucial along the path. A big thank to you, Nako, for organizing this discussion, and to all of you for taking the time and listening to my talk.
Thank you so much, Morteza. Super interesting to see what you're working on now. Currently, I don't see any questions in the chat, so maybe while we're waiting, I can start. It was really interesting to see how different pore sizes affect the different ways in which the cellular material grows or functions. And you said that 200 nanometers was optimal, but can you make pore sizes that are all 200 nanometers, or is it still a distribution? I guess how can you tune the pore sizes precisely?
Exactly. That's actually quite true. There's always a distribution of the pore sizes, and depending of the technique that one uses, the pore size distribution can vary by quite a lot. In the case of the aluminum oxide, nanoporous aluminum oxide that I was using in the beginning of the research, the pore size distribution is quite narrow. They are produced by the anodization process, and they are quite sensitive with the voltage and the electrolyte that are being used in the anodization process. What happens, the pores react to, it's the electrochemistry process, so it is actually quite accurate to make pores within the same size. It was, however, a bit more challenging with the polymer structures that we're creating, because the process behind creating the nanopores in the polymer is to use porogen. So the polymer in the beginning is a liquid, and then we add components to this liquid, which later on can be removed. And once they are removed, what remains is a pore instead of the material. And by having a good control over the porogen size, one could achieve a good distribution of the pore sizes in the polymer structures too. So now we can achieve that range that is satisfactory for our application, for example. At the same time, there is a lot of room for improvement, and it's good to have pore sizes that are more or less within the same range, but also we should keep in mind that T cells also, it is not really the only 200 nanometer exact pore size that the T cells would react to. There is also a range of pore sizes that is around 200 nanometer that the T cells can get activated by entering the nanopores. So it gives us a little bit of room to work with a wider distribution of the pores.
Mm-hmm. Great, thank you. And now we do have a question in the chat, which is, does it require any surface treatment for this process? For growing and peeling off, is it different? I'm guessing on the surface of your nanostructures.
Very good question. No, there is no treatments required. It's a very simple process. Cells start to make those protrusions as soon as they are placed on top of the surface structure. One thing that could be quite critical in this process, that the pores should allow the transport of the water. They should be philic. They should be hydrophilic, and if the water gets into the pores, then that means the cell can also enter inside of the. But there is no surface chemistry required. Although one of the hypotheses that we have, and now we are testing it, is that by changing the surface chemistry, the kinetics of formation of this protrusion changes, and by changing this kinetics, it's possible to change the activation state of the cells as well.
Mhm. And currently it's for immunotherapy purposes, and so you just want optimal growth of the T cells rather than mimicking a more native environment.
Exactly. At the moment, it's if you consider that all the steps are going to be done outside of the body in vitro, it's not maybe too necessary to create an environment which is similar to the native state, which is in vivo and in the body. What we have in mind, though, is that one day it would be possible to inject these T cells and produce these T cells inside of the body. You might think of it as an implant that you place it inside of the body, and it modifies the cells in the body, even help them to proliferate. And if they get exhausted, for example, they can come back, reinforce themselves, and then go back to fight against the cancer. So this would be a more advanced approach to say for immunotherapy.
That sounds like a super cool, like coming back to Starbase before going back to war, something like Star Wars. It's super cool. So thank you so much, Morteza. It was super nice to hear your talk. I think we have no other questions on the chat, so maybe we now move on to our last speaker of today, Dennis. And now I will share my slide. One moment, please. And so I would like to introduce Dennis. Dennis Garoli is a senior researcher and team leader at the Italian Institute of Technology, or IIT, in Genova, Italy. He received his PhD degree in space science from the University of Padova in 2008, and he has 2 bachelor's degrees in both physics and biotechnology from the same university in 2003 and 2011. Since 2014, he is a researcher at IIT, where he is now coordinating 3 European Horizon grants focused on single molecule detection plasmonic platforms for DNA nanotechnology. He has published over 90 papers in numerous high-impact journals, including Nature Nanotechnology, ACS Nano, Nanoletters, the list goes on, many great journals, and his research group is currently focused on the development of nanopore technologies for single molecule sequencing and DNA data storage. Moreover, he is coordinating several international collaborations in nanotechnology and plasmonics applied to DNA nanotechnology. And I met Dennis through a collaboration, so also very super happy to have him today. As of April 2023, his research has been cited more than 2,600 times, and he has an H index of 29. So with that, the title of today's talk is Enhanced Optical Spectroscopy for Single Molecule Detection with Plasmonic Nanopores, the Challenges and Prospects. So thanks a lot, Dennis, and the floor is yours. And I think you're still muted, Dennis. We can't hear you.
Okay. Now?
Yes, now it's perfect. Great.
Okay, sorry. So thank you for the introduction. So my talk is about nanopores, in particular plasmonic nanopores. And so just let me start with a motivation. So our motivation is related to single molecule detection and sequencing. The goal for the community is to define a new technology for single molecule sequencing, in particular for DNA. And this now is something that has been somehow demonstrated as a technology. But this want to be a platform also for protein sequencing. So nanopore is a very simple concept because you can consider a membrane, so an aperture that connects to side of a chamber. And there you can apply an external potential. And if you have a charged object that can be a molecule or a nanoparticle, this can be driven by the external potential. And if the molecule has a size comparable with the aperture in the nanopore, you can detect the event of translocation and maybe also read the information content inside the molecule. This is something that has been explored since the last 20 years for DNA, and now we want to implement the same approach to protein. For protein, the story is completely different because while DNA is made by four different letters, so the four different nucleotide, a protein can be made up to 20 different letters, and this means that if you want to decode the information inside the protein, you need a technology that enables to read 20 different levels, and this is very complicated. This is the representation of the state of the art, both from the commercial point of view and the technological point of view for single molecule DNA sequencing. And is the technology developed and used at the moment by Oxford Nanopore company, that is a billion worth company in the UK. And the technology is very simple. So they use a biological nanopore that is a transmembrane protein with a very narrow aperture. And translocating the molecule to the pore, you can read an electrical signal that can be related to the information inside the molecule. So you can have 4 different levels in the current that can be related to the 4 different letters in the DNA molecule. For a protein, the story is different because, again, you need to read 20 different levels, and this is extremely challenging to be done with electrical recording. For this reason, the community is exploring alternative approaches, mainly based on engineered nanopore, and a very interesting family of nanopores that can be easily engineered in several different ways are the solid-state nanopore. Solid-state nanopore, the most simple example is a silicon nitride thin membrane where you fabricate an aperture. This can be done with different strategy using nanofabrication technique. The interesting aspect is that this is the starting point, but this can be extended to different design. You can fabricate them as an array. You can modify the system with particular coating that can be metallic or dielectric or biological materials in order to really engineer the properties of the nanopore. Now solid-state nanopore is a huge topic of research. The most important field is still sequencing, but there are many others that are explored at the moment. And we are interested in DNA sequencing, protein sequencing, and also, for example, in using nanopores for data storage. Anyway, we are playing with a particular family of solid-state nanopore that is represented here. Is called a plasmonic nanopore. Plasmonic means that you need to integrate in your solid-state nanopore something that has plasmonic properties, and it means that you need some metallic nanostructures. And why this is important? This because a metallic nanostructure with the right design, etc., enable to play with the electromagnetic field and to, for example, to confine the field at the nanopore. And the field confinement means that there you can, for example, implement enhanced optical spectroscopy or other phenomena. And the final idea is to complement the electrical readout with an optical readout. So in order to increase the ability of the pore to read information inside the molecule. So probably the So the first example of a plasmonic platform for single molecule sequencing is represented here. It's a very important paper that now is 20 years old, but is the starting technology for another important company in the field at the moment. The concept is extremely simple. It's just an aperture in a thin metal film. And if you design in a correct way this aperture, you can confine the field, the electromagnetic field at the bottom interface. This means that you can collect the emission only from emitter, so for example from a fluorescent dye or a fluorescent molecule at the bottom interface. So if we go to the real representation of how this platform works for sequencing, this is the representation. So you can fix, for example, a DNA polymerase at the bottom interface, and you translocate the molecule, the DNA molecule inside the aperture, and you perform, for example, real-time duplication of the molecule by using labeled nucleotide, and you detect the burst of 4 different colors during the reaction, just because this platform enabled to confine the field, the light, just in a small volume. Obviously, this is not an optimized system, because here the plasmonic effect is quite small, but you can play a lot with the different design, for example, including plasmonic antennas or alternative design, in order to enhance the plasmonic effects, to enable better signal to noise detection readout in the fluorescent recording. And the final idea is to fabricate and to play with something like this. So is a plasmonic zero mode waveguide where you integrate different nanopores in order to perform the same reaction in a translocation way, in order to have both the electrical and the optical recording. So we played a little bit with this zero mode waveguide configuration, just to demonstrate that if you modify and optimize the design with alternative materials, in particular with gold, that is the most used material for plasmonics, you can enhance, for example, the fluorescence detection by a factor of more than 20 in different geometries. But this is just the starting point of what you can do with plasmonic. So again, the idea is to have a system like the one represented here. So you have the zero mode waveguide with this plasmonic system. You fabricate the nanopore and you make the molecule translocate through it. The interesting aspect is that this system can enable, for sure, to enhance the fluorescence detection, but you can also have additional functionality. In particular, the plasmonic field can be used to generate local heating at the nanopore, and this means that you can modify the properties of the conductance of the pore, and you can also have an optical force that can be produced by the gradient of the field, and this is extremely important because an optical force means that you can somehow trap the molecule inside the pore or some object inside the pore. And in a solid state nanopore, the translocation velocity is a huge issue because typically the event of translocation lasts for a few microseconds, and in these few microseconds you need to read a lot of information. So you want to reduce the speed of translocation. So the first example reported in literature on so a complete example of a plasmonic nanopore to implement both electrical and optical readout is represented here. Is a very simple, again, a very simple platform where you have just this zero mode waveguide with this plasmonic material with some enhancement, and you can perform electrical and optical readout at the same time. This is a demonstration that this is possible, but obviously it is still not a sequencing method because you can read just 2 different colors in a DNA molecule. This because this is not an optimized structures. What you want to have is to to improve the properties of the nanopore, and the best strategy probably is to modify the pore going towards a nano antenna configuration. So the most simple approach for a nano antenna, plasmonic nano antenna, is a dimer as represented here, and you can fabricate this dimer in order to have the nanopore in the gap, and in this way you can really obtain a huge enhancement where you can detect the translocation, and you can also obtain phenomena where you have local heating, as mentioned before, and you can also have optical trapping. So it means that during the translocation of a single object, you can somehow stop the object there or reduce the velocity of translocation, inducing some enhanced interaction between the object and the nanopore. And this has been demonstrated, for example, with a nanoparticle, where you during the translocation, you can trap the nanoparticle in the cavity, and you can perform enhanced spectroscopy with an important integration time. So instead of having just the interaction for a few microseconds, you can keep the particle or the molecule inside your cavity for maybe also for seconds, and this means that you can really collect a lot of signal and a lot of information. This has been also proposed in a real sequencing approach. So this is just a theoretical demonstration where you use these plasmonic effects in order to perform a step-by-step translocation of a DNA molecule, and you can use a particular label-free spectroscopic technique that is called Raman spectroscopy in order to read the different letters along the molecule. So our research is focused now, so a part of our research is focused now on the development of plasmonic nanopores in order to explore alternative spectroscopic method towards single molecule analysis. So the major approach are based on Raman spectroscopy, that is a label-free method, and fluorescence method. For example, fluorescence enhancement, so it's a standard fluorescence, and the FRET fluorescence spectroscopy that is based on the energy transfer between two different dyes that interact in a close proximity. The typical design of our nanopore are represented in these figures, and we are playing with this 3D configuration because we can have with this configuration an important additional functionalities that is related to the integrated nanochannel that you can fabricate. So we can have a nanochannel integrated with our nanopore. It can have some enhancement at the aperture, but we can fabricate this object with different geometry in order to play a lot with the field. So the most simple example again is just a nanopore fabricated on a gold film. What you can demonstrate in this very simple platform is that for example, if you want to play with Forster's resonant energy transfer, so with this FRET spectroscopy, you can, for example, decorate your nanopore with one of the two dyes and make the other one translocate inside the nanopore and detect the interaction with the two fluorophores. And you can demonstrate that this interaction can be modified by the plasmonic effect, by the plasmonic field. You can enhance this interaction, and you can achieve almost single molecule detection with enhanced resolution. The next step was to go towards the third, to the 3D configuration. So the most simple, again, we started from the most simple configuration. This is again our nanochannel, represented here by a dielectric tube. And on the top of the tube, we fabricated this metallic ring. And the metallic ring enabled to confine the field inside the pore. And obviously, if you reduce the size of this ring, you can increase the confinement and increasing consequently the electromagnetic field. And we demonstrated that with this very simple platform, we can functionalize the platform with different fluorophores, and we can make these fluorophores interact in a FRET configuration. We can demonstrate also that we have important enhancement in the fluorescence emission, in the FRET energy transfer. And we also demonstrated that this particular platform can be used, for example, to trap a nanoparticle, trap and release a nanoparticle in a very easy way. But anyway, this is not an optimized nanopore, because nanopore wants to have a size comparable with the size of the single molecule we want to detect. So we want to move from this top design to the bottom one. And to do it, we played with this interesting method reported a few years ago. This paper is interesting, but also very simple. So if you have a plasmonic platform, and you excite this platform with light, a specific wavelength, you can excite the field, and this field in this field configuration somehow can be used to drive also a chemical reaction. And in particular, in this paper, it has been demonstrated that the electromagnetic field confinement can drive the reduction of a silver salt in this nano-hole array. And we wanted to demonstrate this in our nanopore. So in this case, you can see that the position is good, but you cannot achieve a few nanometer of final size, etc. On the contrary, here we used our 3D design in order to go from increasing the exposure time, illuminating the system. We can reduce the size of the pore down to, according to our measurement, down to less than 5 nanometer. And how it works is very simple, because you just need to put your system under a microscope to illuminate everything with light. You drop your metallic salt on top of the sample, and you obtain this controlled reaction. The interesting aspect is that if you look at the electromagnetic field, so consequently at the plasmonic properties, you start from this metallic ring where you have the field confinement inside the ring, but if you then reduce the size of the ring with this photochemical reaction, you can have a much better confinement of the field down to few nanometer as you want for a single molecule detection with a nanopore. This works pretty well also over large array. So it means that you cannot not only fabricate a single pore, but you can also fabricate thousands of them in a quite controlled way. So you start from a large pore and you attain this very narrow pore on all over your sample or all over your chip. And it's also important to stress that this is a localized reaction, so it's driven by the plasmonic field. So you can demonstrate that the deposition happens just over your structures, not all over the sample. And another important aspect is that if you perform increasing the same reaction with increasing duration, you can demonstrate that the rate of growth of the metal is extremely slow. And for example, we measured a metal. The velocity of the reduction in size of the nanopore is something about 1 nanometer per minute. And this is very important, because if you start, for example, from a nanopore of 50 nanometer, for example, and you want to decrease the size down to 5, you can do it in 45 minutes, but you can also stop the reaction in order to have a pore of 15 nanometer, of 20 nanometer, or whatever in the middle. And this is important because maybe your nanopore wants to detect not only a small molecule, but also you can detect nanoparticle or other object that can have at different sizes. And in fact, we demonstrated this platform both for the detection of DNA and so playing with a very narrow pore with a size of 4 nanometer, and preparing another pore with a size of 40 nanometer, so stopping the reaction before, with this controlled duration. We also performed the detection of a mixture of nanoparticles with different sizes. And again, this is a plasmonic nanopore, so electrical recording is important, but is not the final goal of the platform. We are more interested in optical recording. So we performed and demonstrated that in this system you can perform a single molecule DNA detection by means of Raman spectroscopy. And we compare the performance of the initial ring with the performance of the reduced nanopore, and we demonstrated that you have an important enhancement in terms of what you can see as features in the spectrum, and we can also perform this in array. Okay, so Raman spectroscopy to me is very interesting for single molecule detection in nanopore, because is the most powerful spectroscopic technique to perform multiplexing, and is a label-free method. So there are several examples in the literature where you can find the spectra of single molecule through nanopores by means of Raman. The interesting aspect is that every single molecule has its own fingerprint, and it means that also every single building block of a molecule has its own fingerprint. So the 4-letter of a DNA molecule has different spectra. The 20 amino acid has different letters. 20 amino acid 20 different spectra, and you don't need to label the molecule. So we demonstrated that Raman is a very powerful technique to perform single molecule detection towards sequencing, actually. And in order to do it, we played with this very simple platform. So again, is a plasmonic nanopore, so just a pore is into a gold layer prepared on a silicon nitride membrane, and we translocate this metallic nanoparticle that actually is not a spherical particle, but is like a nanostar with different protrusion. And using the trapping effect generated by the plasmonic field, we can trap the particle inside the pore, and this trapping enables to generate the configuration represented at the bottom right figure here. So the nanotip of the nanoparticle goes in the close proximity with the wall of the nanopore and generate this field, huge field confinement that can be called a nano cavity, and the size of, or better, the volume of this cavity is comparable with the size of a nucleotide or an amino acid, because it's close to 1 nanometer cube. Okay, and if you and consequently, if you decorate this nanoparticle with your molecule, you can perform enhanced spectroscopy inside this cavity. And this is actually what we did. We demonstrated that this system enabled to read and to detect the different fingerprint, so the different spectra related to the different, for example, to the different demonstrating that we have a single nucleotide resolution if we decorate the particle with a DNA molecule. And we did the same also with a short peptide, so with a short part of a protein, looking for the different spectra of the different amino acid. And we demonstrated again that with this system we can detect and discriminate, for example, between two different peptides that are very similar, but with just 2 amino acids in different amino acids in the sequence. Okay, so to conclude, now we are moving towards an alternative method where we want to replace the optical excitation in order to trap the object inside the nanopore with an alternative stimuli that is in this case is generated by a magnetic field, moving from this pure plasmonic platform to a magnetoplasmonic platform. A magnetoplasmonic platform means that we can have both electromagnetic and pure magnetic properties. And as demonstrated here, we can trap our nanoparticle, and we can again generate this nanocavity where we eventually can perform enhanced spectroscopy with huge force, much higher with respect to the force that you can obtain with just with light, just using an external magnetic field. Okay, this is just to conclude, and thank you for your attention.
Great. Thank you so much, Dennis, for your really nice talk. Currently, again, I don't see any questions, but maybe they come up. So perhaps I'll start. You talked about the being able to use SIRS to identify different amino acids, but does it depend on the orientation of the peptide? Because I guess the SIRS signature will create different peaks based upon the different way in which the molecule goes through the pore?
Yeah, absolutely. And actually, it has been important to investigate how the orientation of the peptide or also the single amino acid is on the inside the cavity, and you can simulate this different orientation and what happened to the spectra. Okay, but what you see in the spectra is that you have some shift in the peak, but in almost the case you can identify this amino acid. The advantage to you in using a trapping configuration is that there you can keep your molecule for a very long interval of time, so up to minutes if you want. And this means that you can integrate a lot. You can be sure that your spectra is not fluctuating because the molecule interact for a few microseconds or milliseconds.
Mm-hmm. And regarding the trapping, how challenging is it to trap the molecule? Because also, I guess you have to have a low concentration so you can trap one molecule. And so are you diffusion limited to put it in the right place?
No, to trap a molecule is quite challenging. Narrow cavity, so a very narrow, in the case of nanopore, a very narrow nanopore. And it could be quite challenging. The approach we are using is to attach the molecule to a nanoparticle, and to trap a nanoparticle is pretty easy. Mm-hmm. A dielectric nanoparticle, a metallic nanoparticle can be trapped in a nanopore in a pretty easy way.
Mm-hmm. So there's another question in the chat now, which says, "Wonderful work. Can you give more details on the magnetic materials and the process?"
Yes. So the idea here is rather simple, because instead of having just a gold layer and a hole through this all gold layer, you add a interlayer, so you put an additional layer of a magnetic material in the middle. So it's not actually in the middle, but is represented here. So in this case, we used cobalt, but you can also do the same with nickel, for example. And thanks to the properties of the magnetic material, you can obtain this electromagnetic field confinement acting on the magnetic field from the, as a stimuli, not using light, like in the case of the standard plasmonic nanopore. The concept is actually the same, where you just replace the the electromagnetic field from the light excitation with the magnetic field from the magnetic excitation.
Great. Well, thank you so much. I think with that, we'll conclude the talks. And now I think we are going to bring everyone back together and to conclude today's session by maybe discussing a couple of questions that I might have. So I will now share my screen again. Is this? Nope. One second. So we had talks today from Radha and Morteza and Dennis, but I think we are, now we're all on stage. I always have troubles seeing where we are. Great. And so thanks a lot, everyone, for your really exciting talks. Maybe out of curiosity, I was just wondering how everyone kind of got to these very niche fields, whether it's from, angstrom channels to these nanopore biosensing, nanoporous materials. So whoever wants to start, maybe a minute on how you got to this really exciting field of yours.
Right. If I may start,
Sure, please.
Say this would the making of Angstrom channel was completely out of mismatch of me being in condensed matter physics group. So initially I moved to condensed matter physics group as a Marie Curie fellow, and condensed matter physics group was mostly on electrical measurements of graphene, and there was heavy graphene research. So whereas I didn't have any background of graphene electrical measurements or low temperature setups, so I wanted to do what I can do, which was like I was good at fabrication. That was my background. Nanolithography was my background. So it started as a curiosity based, like can we make this as a channel, nano channel, and see if it works. So a lot of time I went in like believing that we are able to make channels which are open. So we thought they were clogged, they would be clogged. Initially when the first paper we said monolayer channel is not possible to make. Because we couldn't detect at the time the flows because of the detection limit, but we thought it was clogged because of hydrocarbons, and that's what also molecular dynamic simulation said. So we said in that paper we said there was no monolayer flow in the monolayer channels. So I would say like the reason we made these channels was curiosity, like whether we can make this kind of empty space.
Really great. And I think it's really fascinating that just depending on where you go and your environment, it really can shape the directions of your independent research. Super. Maybe Dennis, you unmuted yourself. So
yeah, in my case, it's pretty simple because, I have this double formation both in physics and biotechnology. So I really wanted to do some biotechnology stuff in my research. So sequencing is something really in the middle. So sequencing with nanomaterials. So
great. And maybe Morteza, if you have a thought or two.
Yeah, sure. I was generally very interested in nanopores. It was a topic that I started from my master's studies, creating nanoporous aluminum oxide membranes. And it continued during my PhD, during my postdoc in different forms of it. For example, when I was at ETH in the Laboratory of Biosensors and Bioelectronics with Janos Soros, I was more interested in the single molecule sensing with the nanopores, and single nanopores was my interest. And then my interest was slowly shifted towards more biology questions because we were looking at the cells, for example, what is secreted from them, what is inside of them. And at that time, everything was very new to me. I wanted to learn more about it. I put myself in an environment where there were more biologists around me, so I could learn around a lot from them. And then I kept testing my nanoporous materials with different things, and it was a great moment of discovery when I put the T cells on them, and under the microscope, I could see all those bright dots. It was a little bit of a question in the beginning, but then when I realized that T cells like to get inside of the pore, was a moment of excitement, and then after that, it took us years to understand what are the consequences of such protrusions formations.
Mhm. Great. And I think as also someone who works in the nanopore area now, something that I definitely find is a challenge in our field is really understanding mechanisms at these really small scales, and also sometimes reproducibility of these, tiny pores that from angstrom level channels that Radha showed to Dennis's less than a few nanometer pillars. And so I was wondering, what are some of the challenges that you foresee and some of the directions in which we can go to tackle these issues?
Maybe if I can add a few words. So as you already put it nicely, Nako, so this fabrication on reproducibility of these systems is a challenge, right? And the device yield is a challenge. I mean, sharing probably protocol, like maybe having protocol papers on the methods of preparation of these kind of complex. I mean, they may be simple for a group which is always doing it, but not for somebody new who wants to do it. Could be helpful. And I mean, in our own research, what we had done was, as you can see, the use of nano, the angstrom channels was in diverse fields. That's because we went to several groups and collaborated with several groups who are experts in altogether a different regime. So even with IPFL, Professor Alexander Radonovich, who is expert in single molecule imaging, so this was also kind of helpful for us to learn from each other, collaborating with groups who are good at some completely different things. So that was quite helpful. Maybe that's what if I can say, yeah. Thank you.
Thank you.
Yeah, if I can add something, I agree, and I think that the one of the most critical point also now with nanopores is that if you look at the literature, you can find a lot of data at the moment, but if you try to replicate the experiment, is something that maybe you can succeed or not. It depends a lot on how you do it. And this because there's no standard procedure, and it also is also, as a standard approach, you can hardly find the real details on how they prepare the sample and how they activate the surface and how the measurement is really done, or how the measurement are reproducible or not, etc., etc. So this is at least according to my experience, is a huge limitation.
I also think reproducibility could be an issue, but I'm quite positive with the progress in the field in general, because just looking at how things progressed over time, from early studies where only few groups could make nanopores, that now it's pretty much accessible to everyone. And the accuracy is also improving. I'm quite amazed, for example, but what Radha is doing, just making nanopores with 2 layers of water. It's amazing stuff. And I think at some point we would have really the control over the dimensions of the nanopore in a more reproducible manner as the field is progressing. For me personally, or in our research, we are not working with devices as the others are working, but for us, what is important is when we create these nanopores in the substrate, more or less in a bigger picture, we can produce same material that the cells face. So batch to batch, we want to maintain the reproducibility of the material.
Great. Thank you all so much. And maybe a final question before we close out, since it's a young scientist seminar series, although we're all at different points in our career. So maybe 1 or 2 lines of advice for other, students who are interested in these topics of how they can get into it, and maybe how to encourage them to pursue these kinds of tough research questions. Maybe we can go in the same order again from Radha, Dennis, and Morteza.
So it is, I would say these are tough research, as you already put it nicely. These is not easy research. Making the samples, characterizing them, every bit, is not very easy research, right? So, and there could be some sample to sample variability, and there's always, like we are talking about, some sort of statistics, but these are not hundreds of samples, right? So making each sample takes time, and making the measurements also is not easy. Analysis is again a different, analysis is also something difficult. Sometimes you have to develop your own methods, right? So depending on your measurement. So I think if one joins these kind of fields with expectation that they will have to learn a lot of new things. There may be some literature, but they may be developing new things, more new things. With that expectation, if they enter, probably they will feel it rewarding. So I think that expectations is important to know before you enter the field. But otherwise, this field, generally nanopores, nanochannels, and the sequencing, these are, I think, frontier fields now. There's a lot of research and several groups working on these topics. So it's an exciting time to enter these kind of research topics now. So, and there's a lot more to explore, and a lot of capabilities exist now in terms of both characterization, measurements, fabrication. So capabilities exist. It's the right time, but just set your expectations right before you enter the field. It's not easy research. Yeah. Thank you.
Yeah, again, I perfectly agree with Radha. And well, in my opinion, an important aspect to consider now in nanopore is that some applications are more interesting than others. In particular, DNA sequencing is now really inflationated. So because it has been demonstrated already, and so it's not interesting anymore. There is a quite large community that is trying to demonstrate protein sequencing, but in my opinion is really interesting to consider not just the single molecule application, but many others, because for example, we discussed today of this ion selectivity, etc. This, for example, to me is a very interesting topic, and there are many others. So for in my opinion, also what I try to teach to my students is that you need to find your own way towards this in a specific topic. It's not a good idea to just to follow the flow of many others. Maybe it can pay less in the short term, but it can pay more in the long term.
I would maybe categorize my research as biomedical engineering, which is quite multidisciplinary, and it could be a field that somebody from engineering background or biology background from all different disciplines could really do research. But I think what is quite important is the willingness to learn from the other parts that maybe their background education was not in that part. For example, if an engineer can understand the cell biology, it would help him or her a lot to do a better research. Or if a biologist could learn how the material processing work, or how generally what are the characteristics of the materials, different properties, a little bit of physical understanding, that would also make a great impact in their career. So as Radha mentioned, maybe it's the willingness of learning more and more and stepping into the fields that is maybe a little bit far from their background education.
Great. Well, thank you all so much. I think we really had a nice distribution of topics and backgrounds and discussions. I really personally enjoyed it, so thank you so much. I think to close out this section, something I have to do as part of this youth symposium is to give these certificates. And so this is for Radha Boya from the University of Manchester. Thank you so much for joining us today, and we really, really enjoyed your talk. And the next certificate goes to Morteza Aramesh from Uppsala University. Thank you, Morteza, for joining us, and we've really enjoyed hearing about your nanoporous materials and what you've done at Uppsala since leaving us in Zurich. And finally, the last certificate goes to Dennis Garoli from the Istituto Italiano di Tecnologia. And thanks again, Dennis, for your really nice work talking about the single molecule detection. And I also really liked what you mentioned at the end, that it's not just about single molecules, but there's a lot of other interesting applications that we can do with either angstrom or nanoscale pores. So I really enjoyed everyone's talks. Thank you all so much. And the one last thing I have to do is to say that the next youth talks will be happening on May 23rd, on Tuesday again. This is now the Chinese group for flexible manufacturing for sensors and robots in healthcare. So be sure to tune in. And I think that's all I had to say. So thank you all again for joining us today. And thank you to all of our speakers, and hoping to see you at a future another ICANX youth talk. Thank you to all the organizers of ICANX as well for making this smooth and happen nicely. Bye, everyone. Ciao.
不再是奇迹，不再是幻想，此刻真感觉全世界为我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞。不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。I can。没有什么可以阻挡心中无限的力量。I can， 你也能够像我一样飞越最高山岗。我创造奇迹，我拥有梦想。